Hey folks, how's it going? This is Jason Root, and today's podcast we are doing an article review. This article review is a subject that is near and dear to my heart, the article review is on the genetic approaches for sports performance, reflections of a physiologist. This is from the Gatorade Sports Science Institute in the March 2020 issue. The author is Michael J. Joyner, MD. He is from the Department of Anesthesiology and Perioperative Medicine at the Mayo Clinic, Rochester, Minnesota. My association with biomedical diagnostics is that, I got my master's in biomedical diagnostics, and it was heavily weighted into the subject of molecular diagnostics. However, my intent into getting into the program was to study physical state diagnostics. And why do we have such narrow ranges? We only use narrow ranges as associated with disease rather than broader ranges that we can start saying, well, how are we associated with optimal development in our lives, in our health, in our performance, and and with staying further and further away from disease. So basically, we want to have precision, functional, integrated, proactive, preventative, holistic care. So the goal of this, so everything that we write into this specific, everything that we're viewing on the PowerPoint here, everything that's in black is straight from the article. Everything that is in blue is from me, and everything that is in orange I've looked up and go and inserted in, so to give the reader or listener a better concept of what we're talking about here. So the goal, so in his introduction, the goal of this Sports Science Exchange article is to provide an overview and physiologist's perspective on what genetics and genomics can and cannot do. That it would be with respect to performance training. My perspective is presented primarily through the lens of endurance exercise, including elite performance, because this has been the topic of far more studies than other forms of exercise and nutrition. And what I put in here as my reviewer's note, and that is the reason that endurance exercise is so widely studied is because of the research limitations for other types of training, specifically regarding different variables in training and participants of research. So for instance, if you're going to go ahead and study mobility and fascia, it's very difficult to study the mechanisms of mobility and fascia and we end up with too many variations when we're actually looking at the research. There's too many variables to get an appropriate reading, which is actually why the studies on proprioception, fascia, and stretching are shown to be so inconclusive when those who do this work anecdotally know that we know what works in in our sector, but in medical science and in biology it is sometimes very hard to perform different research because of so many variables and so many things that change. So as opposed to maybe chemistry and physics, which don't really have that change so much. So in sports science, endurance athletics, because we're using a singular exercise and singular variables, it's a lot easier to study program design that way. So that's why some exercise modalities are so much more beneficial in the literature than others. Let's see. Going back to the article, the main ideas highlighted in this SSE are the following. Number one, review of the topic of genetic and complex human phenotypes, with an emphasis on the intellectual and scientific progress facilitated by the Human Genome Project and subsequent scientific approaches. Two, the conceptual basis of genetic revolution in medicine spurred by the Human Genome Project. And number three, discussion of the intersection of the concepts stemming from the Human Genome Project and the genetic revolution in medicine and human performance, with a focus on endurance exercise performance. As noted above, this focus on human endurance performance reflects the prolific research and well-described deterministic physiological characteristics of elite endurance performance athletes, which is, as we talked about before, that's why there's, so much prolific research is done on endurance, because it is singular in its program approach. All right, some definitions we've included in here. A phenotype is the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. Epigenetics, the branch of biology which studies the causal interactions between genes and their products which bring the phenotype into being. Another definition is epigenetics studies heritable changes caused by the activation and deactivation of genes without any change in the underlying DNA sequence of the organism. The word epigenetics is of Greek origin and literally means over and above the genome. All right, back to the article, Genesis, and this is a section he has called Genesis of the Human Genome Project. And right here, straight out of the article, in the beginning, the late 80s, many scientists and biomedical thought leaders felt that it was possible to decode the human genome and gain highly informative mechanistic insight into the causes of complex phenotypes. At that time, the sequence of DNA embedded in chromosomes was thought to be a human blueprint that needed to be deciphered, or code that needed to be broken, because it was assumed that there was very tight linkage between DNA variation and human characteristics, such as height, weight, intelligence, and disease susceptibility. The genesis of the Human Genome Project. The Human Genome Project had a $3 billion budget. The first draft of the Human Genome Project was completed by an international consortium of scientists at the University of California, Santa Cruz on June 22nd, the year 2000. 3 days after that, a private firm called Celera also assembled the human genome. There was then a combined press conference from the White House and in London, England, as then President Bill Clinton and British Prime Minister Tony Blair announced the results. A couple of notable predictions presented by President Clinton and Dr. Craig Venter, one of the scientific leaders of the Human Genome Project, are presented below and show the optimism that was typical of just 2 decades ago. All right, these quotes, Genesis. That was, again, Genesis of the Human Genome Project. So these are the quotes from Clinton. In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer by attacking their genetic roots. Just to offer one example, patients with some form of leukemia and breast cancer already are being treated in clinical trials with sophisticated new drugs that precisely target the faulty genes and cancer cells, with little or no risk to healthy cells. In fact, it is now conceivable that our children's children will know the term cancer only as a constellation of stars. By Venter. The genome sequence represents a new starting point for science and medicine, with potential impact on every disease. Taking the example cancer, each day, approximately 2,000 people die in America from cancer. As a consequence of the genome efforts, that you've heard described by Dr. Collins and myself this morning, and the research that will be catalyzed by this information, there's at least the potential to sports science exchange, reduce the number of cancer deaths. Oh, that was in parentheses up there. Let's see, to reduce the number of cancer deaths to 0 during our lifetimes. The development of new therapeutics will require continued public investment in basic science, the translations of discoveries into new medicine by the biotech and pharmaceutical industries. All right, genetic revolution in medicine. So there's a new section. About the same time the Human Genome Project was concluding, the promoters of human genetics and human genomics foresaw the emergence of a genetic revolution in medicine. Key steps in this revolution would be: 1, mapping the gene or genes associated with a disease or pathological phenotype. 2, developing diagnostic or predictive tests based on genetic mapping. 3, engaging individuals at high genetic risk in preventative medicine interventions. 4, prescribing drugs to treat disease based on the genetic profiles of individual patients. This is called pharmacogenomics. 5, using gene therapy to treat genetic defects that cause disease. 6, using information about genetic risk to develop targeted drugs to treat disease. 7, more recently, it has been argued that application of the 6 concepts outlined above would also lead to cost savings in medicine. All right, so mapping genes. Mapping genes is the next section, highlighted by the first category up above on the slide before. A good example of the optimism of the 2000s comes from a quote in the Wall Street Journal from 2006 from Dr. Francis Collins, who is currently the director of the NIH and at the time was the director of the the National Human Genome Research Institute. Dr. Collins said, "I expect there are about 12 genes involved in diabetes and that all of them will be discovered in the next 2 years. So right here I've put in a definition of mapping genes: a sequence of DNA representing the functional blueprint and evolutionary history of the human species. However, only about 3% of this sequence is thought to specify the portions of our 50,000 to 100,000 genes that encode proteins. Thus, an important part of basic and applied genomics is to identify and localize these genes in a process known as transcript mapping." This is from the NCBI website. Gene mapping, another definition. Gene mapping describes the methods used to identify the location of a gene and the distance between genes. The essence of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. So those are some definitions that might help you out when we're talking about certain subjects going through this, especially mapping. All right, to continue the mapping section, there was similar optimism for many forms of heart disease, hypertension, cancer, and Alzheimer's disease, to name a few. When my colleague, the colleague of the author here, Dr. Nigel Paneth, and I reviewed these sorts of predicted associations between DNA variants and disease, we noted that the common diseases and conditions mentioned above involve hundreds of DNA variations across many gene regions. And these DNA variations are implicated as risk conferring. Almost all pathologic or disease-causing DNA variants have tiny effect sizes, and most are remote from any current plausible biological or physiological mechanism. This led Dr. Paneth to come up with a tongue-in-cheek metric called the Genome Futility Index. To do this, he took the number of authors in this area divided by the effect size for the biggest genetic variant associated with blood pressure. So, yeah, he was kind of joking around, but also was pointing out the futility of what we've seen from what was expected from the Human Genome Project and what has actually been produced. And so he continues to write, in this case, the paper had about 500 authors associated with blood pressure, that is, and considered genetic and baseline blood pressure data in hundreds of thousands of volunteers. In this context, the largest observed effect size was that of a gene variant that might influence systolic blood pressure by around 1 millimeter. That's talking about like if you have hypertension at 140 millimeters in your systolic blood pressure, this says that gene might affect it up or down to 141 or 139. And with 500 authors, that says that, so he goes back to the author here. Thus, the GFI for blood pressure is impressively large at 500 over 1 or 500. So basically they're saying that the effect of genes on blood pressure from this 20 years of studying the genetic causes of blood pressure are the higher the number, the lower the effect. And so that's a pretty high number at 500. And it just kind of shows what they're saying is a futile pursuit so far. So now this next section is diagnostics and prediction. And the hope was that genetic information would improve diagnostics and prediction of disease. And he goes on to say, the simple fact is that for most common non-communicable diseases that occur later in life, i.e. type 2 diabetes and hypertension, traditional risk models based upon routine clinical assessments like age, body mass index, and simple blood tests outperform gene scores. Based upon the number of pathologic DNA variants in assessing and stratifying risk. Adding gene scores to traditional risk scores frequently does not improve the risk assessment much, if at all. Importantly, for conditions like diabetes and hypertension, gene scores do not affect the advice to patients. In other words, the advice for those at risk from gene scores and those at risk from routine clinical assessment remains the same: to exercise more and eat more healthfully. So yeah, this is exactly what I've been saying, is that we really need to, it's much more advantageous to approach things from physical state dependent, diagnostics, and to narrow how we look at those to get precision medicine rather than maybe the genetics. All right, in diagnostics, based upon the underperformance of gene scores and similar metrics, genetics and genomics enthusiasts have suggested the use of polygenetic risk scores. This goes on to say, for most diseases, the magnitude of risks due to behavioral and environmental factors is much higher than the genetic risks. That is to say, there's a clear story from behavioral risk factors like exercise and diet, but no clear genetic story has emerged for a vast majority of cases. Taken together, this means that most people with elevated gene scores will not get the condition of interest, and most disease cases or diagnosis will occur in people with intermediate or low gene scores. So he's saying that the correlation between genes and disease has been pretty low, but the correlation between behavior and disease is very high. In preventative medicine, another key idea emanating from the Human Genome Project and the genetic revolution in medicine narrative is that informing individuals about their genetic risk would somehow stimulate proactive preventative changes in behavior that would limit or reduce the risk of disease. And preventative medicine continued, simply put, there's very limited evidence for this assertion. As a recent meta-analysis noted, expectations that communicating DNA-based risk estimates changes behavior is not supported by existing evidence. Again, we go on in the preventative medicine. Additionally, we need to remember that individuals frequently receive all sorts of straightforward information related to their health. For example, what I have called the bathroom scale score. In spite of this highly informative risk factor, he's talking about what you see on the bathroom scale, it is difficult for most humans to lose weight and sustain their weight loss. In this context, is it realistic to think that a gene score will be more effective than a bathroom scale score in promoting the sorts of long-term behavior change required to successfully modify lifestyle-related risk factors? A noteworthy digression regarding the nexus of body weight and gene scores. In terms of weight loss, current evidence does not support the idea that either allowing people to choose a diet that they prefer or tailoring a diet based on some hypothetical genetic information will enhance weight loss. However, it is known that across a wide spectrum of diet types and interventions, that the main determinant of dietary success in long-term weight loss is adherence to the diet itself. So this goes on to say adherence is way more important than knowing more about genes or anything like that. Anyways, pharmacogenomics, the next section. So the goal is to tailor pharmaceuticals to specify two specific genotypes. And he goes on to say, well, the evidence is so far disappointing. And in cancer care, there is no difference or modest effect size in what's happened so far. Additionally, by 2018, only about 5% of tumors have been shown to respond to targeted therapy. Finally, recent evidence from a variety of sources shows that cancer is a multifactorial disease, and the idea that there is a single simple target that can be identified and used to cure patients is unrealistic. Again, while there are clearly some successes using this paradigm, it is unclear whether the hope for a broad-based cure for cancer will emanate from this approach. And I'll go just a little bit of note for myself. Yeah, cancer is a group of diseases, not really one disease. All right, gene therapy. And so my definition I put in here, the transplantation of normal genes into cells in place of missing or defective ones in order to correct genetic disorders. So back to the author. This is the idea that gene therapy would be used to cure diseases. He writes, he goes on to say, unlikely to be a viable approach for many common non-communicable diseases. However, recently a number of niche successes have been seen. These exciting results for things like blindness, some forms of muscular dystrophy, and other conditions are impressive examples of how biotechnology can be harnessed to cure disease. But then he goes on to say, however, the cost of these treatments is immense, frequently in the millions of dollars, and it is unclear how society at large will afford these products. Next section, drug discovery. A key element of the genetic revolution in medicine narrative is the idea that large numbers of novel drugs would be generated as a result of the Human Genome Project and related technologies. And he goes on to say, there has not been a vast speeding up of drug target identification, validation, and new drugs approved in the genomic era. So from 2000 till now. Cost savings is the next category. A final area where the anticipated genetic revolution in medicine was going to transform the medical ecosystem was via cost savings. The idea being that the combination of screening, preventive interventions, and targeted therapy would make the medical system more efficient. And he goes on to say, the concern on this is that if you follow up on every bad gene variant identified, this will lead to overtesting and overdiagnosis at a scale and actually increased cost and do far more harm than good for the average patient. This means that the medical care system is at risk of succumbing to what has been termed the incidentalome. In this scenario, alarmist medicine which is overscreening, overdiagnosis, and overtreatment, would likely to be a major unintended consequence of the envisioned genetic revolution in medicine. This also makes it unlikely that such an approach will reduce costs. And so a note from me would be one example of this that we studied was Angelina Jolie, the famous actress, had a double mastectomy in 2013, strictly based on her BRCA, or what they call the BRCA gene, identification for breast cancer. And it is very possible that this prophylactic approach to having to breast cancer by having this gene was just simply not needed. So she had a huge procedure that was very possibly not needed for a condition that may never have come. The nexus of genomics and endurance exercise. So now he gets really into endurance exercise rather than the medical or disease part of his article. Now he gets straight into what it means for endurance exercise. As I said, we've seen a lot of companies and a lot of people out there trying to target what this Human Genome Project and genomic medicine can do in terms of performance, and right now he's going to attack that. So back to the author. Although the implications of genetic revolution in medicine are commonly centered on the study of risky genes, equally interesting are the implications for the study of elite phenotypes and elite endurance athletes. The nexus. So this is in the same category. So the central question is, what do we know about the genetics of these factors? However, before we go any further, we need to establish criteria for genetic causation. Here are some elements of how this topic might be considered. Identify potentially causal gene variants. Another is link these variants to deterministic physiological mechanism. Third, explain more than a small fraction of the variability in the physiological responses. Fourth, demonstrate the gene or pathway required to the response of interest. And the last one is show that the conclusions hold up when a maximal adaptive stimulus has been applied. All right, so right here, he's going to be looking at VO2 max, and the definition of VO2 max is also known as maximal oxygen uptake. So we assume that that's what we're really looking for in our endurance athletes. And it is a measurement of the maximum amount of oxygen a person can utilize during intense exercise. It is a common measurement used to establish the aerobic endurance of an athlete prior to or during the course of training. And then, so back to the author, VO2 max utilizes in, or VO2 max values in monozygotic twins, or identical, are highly correlated with less correlation among dizygotic or fraternal twins, and even less correlated between siblings. These data indicate that VO2 max has a heritable component and suggests there's a strong genetic component as well. So this would give the concept that genetic studies may have something to contribute to to this, but we'll go on to see what he says about it. There are several well-defined deterministic physiological mechanisms that contribute to VO2 max. Maximal oxygen uptake is determined largely by maximum cardiac output, along with red blood cell mass, or the highly related total blood hemoglobin. This simply means that to have high VO2 max, individuals must be able to pump a lot of blood from their heart, and that blood must be rich in oxygen. So in VO2, let's see, importantly, a large cardiac stroke volume is a key component of the high cardiac output values seen in elite athletes. Stroke volume is the amount per beat of blood that the heart pumps out. At this time, there are no clear genetic signatures or gene variants associated with stroke volume, blood volume, or total blood body hemoglobin. So although we see some association, this is me right here, although we see some association with the genetic component looking at twin studies, and fraternal studies, we don't see a clear genetic link anywhere to stroke volume, blood volume, or total blood hemoglobin. Back to him. Gene scores for maximum heart rate explain at best a few beats per minute and are thus likely to have much influence on VO2 max. Then we have upstream in the oxygen transport cascade are the lungs. So what's the pulmonary part of this? And again, at this time, studies involving large groups of humans have failed to demonstrate any clear genetic signatures associated with superior lung size or function. This suggests that there is no easily identifiable genetic component to this element of the oxygen transport cascade. All right, now he goes on to talk about other mechanisms from blood vessels, mitochondria, lactate threshold. Blood vessels. There is no clear-cut genetic explanation that account for variability in this feature of the cascade. So blood vessels, if you know, you have blood vessels, that are, the blood vessels are part of the cardiovascular system, and so, cardiovascular return coming from blood vessels, that might have, an effect on, on VO2 max, and therefore, if we can show a genetic relationship, then, we can show how that affects things, but we don't see any of that. Mitochondria. Recreational runners to elite endurance athletes can have similar mitochondrial adaptations to training. Again, it is hard to imagine how small differences in mitochondrial function caused by gene variants would have a large impact on VO2 max. And at lactate threshold, no genetic insights into factors that explain variability in economy or efficiency. So looking at trainability. So basic observation is that in response to the standardized program of training, and I'll put this in research, there is a wide range of VO2 max responses in individuals. The magnitudes of these responses tend to cluster in families, and some individuals have been termed non-responders. He goes on to say, gene variants associated with trainability are remote from the key physiological pathways in the oxygen transport cascade. Next, he says, most training studies reporting there are non-responders to training are of modest duration and moderate intensity. So that's why I mentioned that it was in research before, because by contrast, he goes back to say, when high intensity interval training studies are considered, it appears as if almost all humans are trainable to some extent, and many humans can generate increases in VO2 max far beyond those normally seen with traditional fitness type training. So we see that in the field. This is where I want to note again that this is an example of limitations of research versus what we can do in the field. When you have somebody at a large research study versus what we can do with somebody one-on-one, what we see is vastly different than what the research can show, because of the ability to really do different, to apply different sorts of training methods. Change color of that for you folks out there that are actually watching this. All right, so the summary and practical applications. This is in the article. Why are there no clear genetic signatures for physiological responses to and determinants of endurance exercise? First, in human phenotypes, there is a clear genetic signature linked to the causal physiology. The signature is typically associated with something that might confer a marked evolutionary selection advantage. And so this is where he goes, and I'm just going to summarize here. He goes on to analyze the hunter-gatherer environmental pressures and gains no clear correlation in that on physiological genetic adaptations for VO2 max as they're studying here. So that was just something he brought up as a summary. Also, summary and practical applications, he goes on to say, because performance is multifactorial, and because the genetic underpinnings of key elements of performance remained obscure, it seems like the best way to identify talented individuals is to simply field test them using things like timed runs and measures of strength and coordination. Information from these tests is essentially a summing circuit for factors related to the cardiovascular system, skeletal muscle fiber type, coordination, and biomechanical factors. Because each of these factors alone may have hundreds of genetic determinants with tiny effect sizes of their own and an uncertain relationship with the trait of interest, field testing for athletic ability is likely to remain paramount for the foreseeable future. And so this is where I was saying state-dependent test, whether on the performance or whether we're talking about disease markers, you know, in blood or anything else, and continually looking at how each of these state-dependent tests shows factors is, you know, paramount importance as opposed to genetic testing. The key points in conclusion. Now, I want to note in the article, he actually put these key points in the beginning of the article, but I thought for this presentation they would be better at the end. But these are his key points. So in general, complex human traits do not have simple genetic explanations. Second key point, this is true for common medical conditions like hypertension, diabetes, and most forms of cancer. It is also true for things like exercise capacity. So I put in here, it's true for disease, fitness, or high performance, all equally. Next point, for sports scientists, this means that it's going to be hard to use DNA to predict who is going to be good at what and tailor training programs to individual athletes based on variations in their DNA. And the last one is field-based testing for athletic potential is likely to remain the most powerful tool to assess talent for the foreseeable future. And I would add on to that, you know, on the lower end of things, we're talking about disease, still state-dependent in actionable ways is paramount. So root health conclusions. So we're going to go into my conclusions on this, and that would be for health and disease. For health, disease, and performance, all equally, we need more attention directed at the healthcare system for more actionable or physical state diagnostics and less spent on molecular or genetic testing. That, you know, that's, you know, if we have the same amount of money and we're going to take some money, put it towards genetic, some money, put it towards, you know, state testing, then, you know, I'm all for putting a majority of that towards physical state diagnostics. My second point, this should be done with an emphasis on lifestyle, training, nutrition, and human environmental re-evaluation. And what I mean by that is making things in our environment actionable. Very hard to make things, you know, if you don't have parks and sidewalks and things like that, you know, it's hard to be actionable towards, you know, your lifestyle. In terms of performance, there's a plethora of new functional and performance testing for high-level individuals. So, yeah, we need to keep on going with that. And I love what's being come up with out there in the marketplace. You know, for trainers, kinesiologists, physical therapists, chiropractors, all along, you know, those sorts of professions. And I am a huge fan and think that the effect could be so much more if the general public were also open to engaging in programs that are based of these tests. So I'm a huge fan of these tests. So we need to get the public more, we need to concentrate more on getting the public engaged in these tests through education. You know, the old physical fitness test, presidential physical fitness test was a wonderful thing, you know, and yeah, we still follow that to this day, but even taking that and, you know, expanding on that is a great idea. So if you contact information, if you'd like the PowerPoint on this or any more information on root health, contact me at my phone number that's listed here, 310-922-3849, or at the email rootfit@gmail.com. And you can look at Gatorade Sports Science Institute to find this article. It's pretty recent, so you should be able to find it pretty easy. So thanks a lot, folks, and for joining me today. And I will talk to you soon.